Good evening. We're here at the Exeter Cable TV studio, ready to show the second of our three-part series on climate justice for future generations. Tonight's program is called The Secret Life of Trees. And before I introduce UNH Professor Emeritus Barry Rock, I'd like to just let you know that this whole series is produced with Bob Glowacki's help at the Exeter TV studio, produced through the efforts of the First Unitarian Universalist Society of Exeter, otherwise known as the UU Church in Exeter, and the Social Justice Committee of that church. And it's through the generosity of a grant given by the late Ingeborg Locke. It's an opportunity grant that allowed us to put this program together. And it's supported by 350NH, the Water Street Bookstore, and the Exeter Tree Committee. So tonight's program is going to be given, it's actually a recorded session. We recorded the sessions several weeks ago. Professor Barry Rock recorded them then, but he's here in the studio live, and we're going to take questions. So if you have any questions, put them into the Q&A, and we'll try to address them as the program goes along. Professor Barry Rock is a faculty fellow and scholarly coach, research and engagement academy scholar coach in the Writing Academy, and he's a professor emeritus from the Department of Natural Resources and the Environment at UNH. Barry grew up in Vermont, and he came to UNH in 1987 after getting a biology degree at Alfred University in New York. Associate and professor of biology at Alfred University. He got his BA in botany from the University of Vermont, got his master's and PhD degrees in botany from the University of Maryland, and then after he got his doctorate, he joined Alfred University. Dr. Rock became the site botanist for the joint NASA GeoSat remote sensing study conducted at Lost River, West Virginia. And then, as I say, he joined the faculty at UNH in 1987 as an associate professor of forest resources and at the Institute for the Study of Earth, Ocean, and Space, otherwise known as EOS. Dr. Rock's research and publications have focused on remote sensing of vegetation, specifically on basic and applied research dealing with biophysical properties, such as pigment concentrations, anatomical characteristics, moisture conditions of leaves, and their influence on reflectance features, which may be remotely detected to identify problems with the plants. During the 94-95 academic year, Dr. Rock assumed the position of senior scientist and assistant director of GLOBE, which was the Global Learning and Observations to Benefit the Environment program, which was part of environmental education outreach project directed from the White House. Dr. Rock developed a hands-on science activities program to be conducted by GLOBE students on an international scale, with 26 participating countries involving over 2,500 schools. GLOBE was patterned after ideas presented by Al Gore in Earth in the Balance. Between 1997 and 2001, Dr. Rock coordinated a New England regional survey of climate change impacts to the region, both over the past 100 years and those projected for the future. He does have a curriculum vitae that goes on, but let me just say that he has worked with National Science Foundation, NASA, NOAA, and the Fulbright office. And he was at the NASA Jet Propulsion Laboratory in Pasadena, the NASA Goddard Space Flight Center, and at the Charles University in Prague, where I visited in the past. So, Professor Rock, The Secret Life of Trees.
Thank you, Herb. It's a pleasure to be here tonight. I look forward to hearing from our viewers what they find out about the secret life of trees. I think most of us think of trees as passive bystanders. They're there, but not doing anything. I think as we learned last week, there are a lot of things that they're doing, especially related to climate futures, absorbing a tremendous amount of carbon dioxide, just doing what trees do. So I look forward to any questions anyone might have.
Wonderful.
Thank you. Good evening. Tonight I'd like to talk about a topic that's near and dear to my heart. I'm a botanist, but at the University of New Hampshire, I have taught forestry-related courses for 25 years. And the topic tonight is going to be the secret life of trees. I've spent 30 years of my career studying forests, and in particular forest health. And I use Earth-orbiting satellite data to look at forest health around the Northern Hemisphere. So we'll be talking about the forest health and that sort of thing, but tonight we want to start off with the secret life of trees. So here's a kind of a summary of the topics. We're going to talk about tree structure, leaves, wood, roots, etc. I'm going to talk about transpiration, the loss of water vapor through stomates from tree leaves, and the role that that plays in cloud formation. You may never have thought that trees had a role in that, but in fact, they have a significant role. We're going to talk about the reflectance properties of green plants and what they tell us about forest health. And that becomes really important. As we'll see later, trees communicate with each other, but they also communicate with scientists and property, the reflectance property. So we'll talk about that. Then there's a fungus among us. Plants have beneficial fungi in their roots. About 90% of the forest trees in the world have fungi associated with their roots, and this allows the tree to take up nutrients and water more efficiently. It's a protection against disease, but it also allows the trees to communicate with one another. So we'll talk about that. And that root network, including the fungi, the Mycorrhizae, is referred to as the wood wide web. So we're going to be talking about that. There are some useful terms that I think I need to introduce. You're probably familiar with these anyway, but just so we're all on the same page. Angiosperms and gymnosperms. Angiosperms are flowering plants. They are the plants that produce fruit. Think of an apple, think of a peanut, and it sets them apart. The term angio comes from the Greek angion, which means a container or a vessel, and sperm comes from the Greek word sperma, which means seed. So angiosperms produce their seeds in a container of fruit. The gymnosperms, on the other hand, the term gymnos in Greek means naked. And so a gymnosperm is a naked seed. And what that means that the seeds are not contained within a fruit, but rather are open on an open pine cone, for instance. So it turns out angiosperms are hardwoods, and gymnosperms are softwoods. Turns out that the term hardwood and softwood really has nothing to do with the hardness of the wood. It's a taxonomic term. So hardwood is an angiosperm, and balsa wood, if any of you have made model airplanes, as I did as a kid, using balsa wood, that's so soft you can just crush it in your hand. I mean, it just falls apart, and yet that's a hardwood because it's an angiosperm. Monocot and dicot, it turns out that angiosperms are divided into two categories of plants. Monocots, which are the grasses, palm trees, corn stalks. And the term monocot just means one cotyledon. It's a seed inside, actually it's the embryo inside of a seed. So monocot, one cotyledon, dicot, two cotyledons. We're going to talk about xylem and phloem. Xylem is essentially the tissue in plants that conducts water and nutrients upwards from the soil to the leaves. And we're going to talk about wood, and wood is xylem. It's just a massive complex of conductive tissue that is designed specifically to conduct water and nutrients upwards. Phloem, on the other hand, is in the bark of trees. It's the inner bark, and it will turn out that the phloem is a tissue that conducts the photosynthetic byproducts, sugars, for instance, and other things downward from the leaves, down through the bark, down to the roots. So I've already talked about inner bark and outer bark. We can mention primary and secondary growth. Primary growth is growth in an apical meristem, the tips of branches. Think of the terminal buds. Within that terminal bud would be the apical meristems. And then you have wood being produced by something called a vascular cambium, and that produces secondary growth. And I'll point things out as we get to some examples of that. And as I've said, wood conducts water and nutrients, and it does that in angiosperms in something called vessel elements. They're just hollow tubes, kind of like this. Gymnosperms, on the other hand, don't have vessel elements. They have tracheids, and those are specialized cells designed for conducting water and nutrients, but they're not open on their end walls. We're going to talk about tree structure first, and let's go to the wood. Some diagrams here showing particular situation on the left-hand side of the screen. We're looking at a schematic of a tree. The apical meristems would be up where the leaves are at the top, and the root tips as well. And the vascular cambium, producing secondary growth, are those different layers, growth rings, if you will, in the wood. I'll use my pointer here, I hope. Well, I won't. On the top image on the right-hand side, we have the outer bark first, and then the inner bark. And remember, the inner bark is the phloem. The outer bark protects the tree from drying out, and in particular protects the phloem from drying out. Turns out that the phloem is about 100% living, and so it's got a lot of cells with a lot of moisture in them. Wood, on the other hand, is 99% dead. That may surprise you, but it turns out there's only one type of cell in the wood that is living. The other cells, the vessel elements, the tracheids, supportive fibers, those are all dead at functional maturity. You see the cambium there between the inner bark, the phloem, and the sapwood of the wood. Notice the sapwood is lighter there. Turns out that only the most recent 2 or 3 growth rings in wood are conductive. That's why it's called sapwood. The darker inner wood is called heartwood, and heartwood is a storage area. So wood actually has 3 functions. One is to conduct the water and nutrients. Second is to support the tree and its leaves that are in a canopy up where it can get to the sun. And then the third is to store useful materials. It turns out a lot of starch, a kind of sugar, if you will, that the tree needs to grow, the vascular cambium especially. and that's stored in the heartwood. We also see in the bottom graphic cross-section, and then radial section and tangential section. Those are just different sections of wood. We're going to be seeing examples of those, so kind of keep that in mind. Here's a cross-section of an angiosperm stem. So technically it's a hardwood. And you can see that in the very center is something called the pith. And that's a primary tissue. It was produced in the apical meristem. The X, you can see the blue circle. That's X stands for xylem. It's secondary xylem. You can see that the cells are in nice radial files. They've been produced from the vascular cambium. Over on the right-hand upper corner, you see we've labeled the vascular cambium. That's between the outermost part of the secondary xylem and then the secondary phloem, which is the inner bark. The P stands for phloem. The BF is not best friends, it's bass fibers, and that's commonly found within the juncture between the phloem, that's the inner bark, and then the outer bark. The C stands for cortex, and then you've got epidermis for the EP. I hope you agree that that's a pretty interesting image. I actually made that myself by cutting a twig with a razor blade. And you can do the same thing at home. Just be careful. This is a cross-section of a conifer with all of the parts labeled. We'll start on the outside. You see the bark, that's the outer bark, and then below that, the inner bark, the phloem. Then we come to the vascular cambium. There's a false growth ring labeled. We're not going to talk about that today, but we will talk about that in our next presentation. You can see that conifer has a very uniform secondary xylem, wood. It's because it's mostly all tracheids. But you also see some little holes there, pores, and those are resin ducts. And of course, gymnosperms are resinous. Think of the nice smell of a pine forest. And then there's an annual ring that's labeled, and then you've got late wood labeled and early wood. We'll talk a little bit about those later. And then, of course, the central pith. This is just a close-up of the outer bark and the inner bark, the vascular cambium and the secondary xylem, or the wood. This is of a gymnosperm, and you can tell that by looking at the wood and the kind of homogeneous nature of the cells there. Those are all tracheids, seen in cross-section. This is a diagrammatic representation of the cambial zone in a gymnosperm. And if we let's start from the inside, from the left-hand side. That's showing us wood. You can see that some of the tracheids there have little circles. They look like donuts or bagels. Those are bordered pits. We're going to talk more about those later. They facilitate the movement of water and nutrients upward in the secondary xylem gymnosperms. The first tracheids there are early wood tracheids because they've got nice, large, open centers to their cells. And then you see they get really tight, and that's the late wood. And then you see the next growth ring is the early wood again. The late wood really hasn't formed yet in this diagram. The vascular cambium is labeled, and then you come to the inner bark or the phloem. Down along the side, you see the radial section. Those are the living cells of the wood. Remember I said the wood is mostly 99% dead at functional maturity. All of the tracheids are dead, but the radial section, those are living cells. We have a similar diagram for an angiosperm. I think if you notice, the big difference is that the angiosperm has these vessel elements, the large circular holes, whereas the secondary xylem of a gymnosperm was all tracheids. And we see the same sort of thing. Some of the smaller cells are fibers. The larger open cells are just like a soda straw, and those are the vessel elements. The cambium looks the same. And you've got the secondary phloem on the outside, and then the living cells along the radial section. Those are the cells of the vascular rays. This is a scanning electron microscope image of a gymnosperm wood. And I just love working with the scanning electron microscope because it gives us this beautiful three-dimensional view of whatever we're looking at. In this case, it's a gymnosperm, again, very homogeneous tracheids in the wood. You see a growth ring, that sort of thing. Very useful being able to see everything in 3 dimensions. You've got the cross-section, and then along the kind of the left-hand side, that's a radial section. You can see some of the vascular ray tissue. This is just a close-up of bordered pits, and it's a pair of pits. 2 adjacent tracheids have these bordered pit pairs, and that's how the water will move through from one tracheid to another and eventually get to the leaves. This is a diagram of the bordered pit pair, and you'll notice under A that this is a typical functioning bordered pit. You've got the dark feature kind of in the center there. That's called a torus. It's waterproof. And when something happens, a disturbance, an insect, a Boy Scout coming along with a hatchet and chopping away at the wood, that torus moves over and blocks the opening. That's called an aspirated pit, and it's just incredible to me that we see this kind of cellular structure, and it's designed to keep the vertical column of water in place if an insect takes a bite out. Instead of the whole thing dropping down, it's stopped because that torus goes over to the side and plugs what's called the pit aperture. This is what they actually look like. I took this photograph. Here we're seeing bordered pit pairs in face view on the left-hand side. And then on the right-hand side, this is a radial section. And you can actually see the bordered pits. You can see the torus in the middle. This is a functioning set of tracheids. Big difference now when we look at the scanning electron microscope image of an angiosperm wood. Instead of just everybody being a tracheid, we've got that mix of vessel elements. Those are the big holes. The smaller cells are the fibers that support the tree and allow it to get to be 100 feet tall without falling over. And here we see a tangential section. You see the cross section, but then the tangential section, and you can see the vascular rays in section here. You can see how they are made up of a number of cells. And again, those are the living cells of the wood. Okay, let's talk about the leaves and the roots. Leaves, of course, their primary function is photosynthesis. You may or may not know this, but there are 2 steps in the photosynthetic process. Remember, in photosynthesis, you're going to have water and carbon dioxide being combined in the presence of sunlight and made into sugars of a variety of sorts. Think of glucose, fructose, sucrose, etc. And in the process of that occurring, making sugars from water and carbon dioxide, the water is split, and that's called a light reaction. And one of the byproducts of photosynthesis is oxygen. You and I are here today because of the photosynthetic process going on around the world. Our atmosphere is largely nitrogen, and then 21% oxygen, and that oxygen has come from photosynthesis. So after this presentation, I want you to go out and hug a tree because they've got incredible work. That's the light reaction. It requires light. In the dark reaction, we see that CO2 then goes through a series of steps, and the end product is sugar. That's called the dark reaction. It doesn't require dark. It goes on in the light, but it's only the light process that actually splits the water and releases the oxygen. Typical diagram of a dicot leaf, an angiosperm leaf. You probably remember this maybe from high school biology. Got the different parts labeled. I won't go through all of that, but I would point out that the center part of a leaf is called the mesophyll. And the term meso means middle and phyll means leaf. So it's the middle of the leaf. You see a stomate at the bottom. We're going to talk a little bit more about those later. This is what it actually looks like. This is a leaf of Angiosperm, a dicot. And again, the parts are labeled. You've got an upper epidermis. You've got a vertically oriented cells called the palisade mesophyll. And below that, just kind of randomly oriented cells called spongy mesophyll. You've got vascular tissue, xylem and phloem. We've mentioned those. You can see a couple of stomates on the bottom in the lower epidermis. And the photosynthesis is going to be going on in the mesophyll, both the palisade mesophyll and the spongy mesophyll. Here is a scanning electron micrograph image of a grass blade. Remember, grasses are monocots. We'll zoom in on it. You almost see little faces there. Those faces that look like eyes are actually the vessel elements of the xylem of that monocot. The sieve cells of the phloem are on top, kind of like the forehead, if you will. And then you've got the mesophyll off on both sides. It's amazing what you can see when you've got some really good magnification. Chloroplasts are found in the palisade cells and the spongy cells. They are little packets of coin-like structures. You see that down in the lower right-hand corner. That's called a thylakoid, and that's where the light reaction is going to be going on. You can see, if you look at the whole chloroplast, kind of a dark gray area that is labeled stroma. That's a liquid material, and that's where the dark reaction is going on. So even in the chloroplast, Mother Nature has split the light reaction. That's going to be done in the thylakoids, and then the dark reaction is going to be done in the stroma. This is an electron micrograph. It's not a scanning electron microscope, but a transmission electron microscope. And those thin membranes are in a stack. It would be like a stack of pennies. And we're looking at it from the side. Those are the thylakoids. And then kind of the granular area around them, that's the stroma. Isn't it amazing all of this is going on in a leaf? Here we see some dividing chloroplasts. One of the theories as to how chloroplasts evolved is called the Endosymbiosis Theory, and that says that chloroplasts originally were small algae. This was before cells had developed. This would be 3.5 billion years ago, long, long time ago, but there were living cells. They were green, they had chlorophyll, they were algae, and they were producing the first oxygen that created the atmosphere that we know today. And the fact that you can find chloroplasts dividing indicates that they probably were organelles originally that could undergo division. There are other pigments as well. This is a pretty picture of some African violets, but the important thing there is those other colors can be indications of stress in vegetation. Okay, transpiration. That's the loss of water vapor coming out of the stomates in the leaves. You can see that. The water enters the plant through the roots. In fact, the mycorrhizal fungi play a big role in water uptake. But water comes in through the roots, goes up through the stem via something called capillary action, and then out into the leaves. And as that water evaporates from the very moist mesophyll cells, that water vapor goes out through the stomates and enters the atmosphere. This just says what I just mentioned. Transpiration involves the loss of gaseous water vapor, from the mesophyll via stomates, and as well as adhesive and cohesive forces that hold the molecules of water together and allow them to move upward, and are being actually drawn upward by the evaporation of water from the leaves. This is another schematic diagram of the same sort of thing. Water entering through the roots, going through a number of cells in the root, going up into the vessel elements or the tracheas of the secondary xylem, and then out into the leaf through the base of the leaf, which is called a petiole, and then eventually out. So it's kind of a big circulation. Remember, the water vapor going out of those stomates is going to condense and form clouds. It's going to rain, and that rain drops to the ground and then enters the plant again. So Mother Nature is a marvelous example in hundreds of ways of very purposeful recycling. It's amazing. And then here's just a diagrammatic representation of a stomate. You can see stomates, the CO2 enters the stomate, gets incorporated into the dark reaction in a chloroplast, but then exiting through the stomate would be water vapor and oxygen. So it's all part of a day's work in a plant, in a tree. Now, this graphic is really interesting. This is the Amazon Basin, and you can see a lot of little white dots. Those white dots are actually clouds, little puffy popcorn clouds, and each of those has come from one or more trees. So like I said before, a large percentage of our clouds come from leaves. And so we're dealing with, you see at the bottom it says it accounts, transpiration that is, for as much as 70% of clouds. So again, go out and hug a tree. It's generating clouds. Of course, it has a rough time doing that in the winter when the leaves are gone. And so the clouds that we see outside on a winter's day, that comes from other processes as well. But during the summer, a large percentage of clouds come from the trees. This is just a schematic diagram of a root cross-section. You can see that you've got an outer epidermis, root hairs associated with that. You can see water molecules coming in, entering the cortex. That's living cells, a lot of starch stored in the root cortex, something called the endodermis, and then the xylem tubes, the vessel elements, phloem, etc. Just marvelous structure. So let's talk about measuring the reflectance properties of leaves. Some of you have probably heard of the electromagnetic spectrum. Energy within our Earth system falls into a large number of different categories of this electromagnetic spectrum. Right in the center, you can see kind of the representation of light. We have radio waves, we have microwaves, you use those in your cooking. You've got infrared, you've got the visible, that's what we can see with our eyes, ultraviolet, X-rays, gamma rays, etc. And all of these are going on all around us, and the only part we see would be the visible. And that's the only part that plants have evolved to take advantage of. This is just atmospheric transmission. It means that our atmosphere is transparent, not transparent, transmissive in the visible, but then at other longer wavelengths as well. And so it's amazing that you and I have evolved vision where you have the greatest amount of atmospheric transmission, and plants have evolved photosynthetic processes at exactly the same spot. So just amazing. This is a typical vegetation reflectance curve. I have dreams of this at night. This has been my career for 30 years, and we can see along the vertical axis percent reflectance going from absolute black to absolute white, from 0% to 100%. Along the horizontal axis are the wavelengths of the visible and then something called the near infrared. It's called that because it's near the visible, and then shortwave infrared. And plants in the visible, if you look above at that curve, you can see they're fairly dark, and that's because they absorb so much light in the process of photosynthesis. The only light they don't use is green, and so you see a little modest green peak there. And then there's a very sharp rise called the red edge, and that sharp rise takes us to the near infrared, and it's beyond where chlorophyll is absorbing, and so plant structure is very light and bright at that point, can be 80%, 90% in some cases. And then we see two water absorption bands that are characteristic of our atmosphere, so there's no real radiation coming from the sun, no solar radiation in those regions. And then you've got two peaks in what's called the shortwave infrared. And you see that Landsat, which is a satellite, has spectral bands, and you see those along the top there, bands 1, 2, 3, 4, 5, and 7. Band 1 is in the blue, band 2 is in the green, band 3 is in the red, band 4 is in the near infrared, and band 5 and 7 are in the shortwave infrared. And each of those bands tells us something about three properties of a leaf. The visible properties, bands 1, 2, and 3, those are leaf pigments, chlorophylls, a variety of pigments. And then band 4 is the near infrared, and that tells us about the cell structure. And we can see this from orbit, which is just absolutely amazing. The fact that from 500 miles up, I can look at a vegetation reflectance curve, and from the near infrared brightness or darkness, I can tell how healthy that vegetation is. And then bands 5 and 7, they tell us how much water is in the leaves, the canopy of the trees. And as a botanist, if you know about leaf pigments, and you know about cell structure, and you know about water, what more could you ask for? I mean, it's just amazing, and we can study this from orbit, Earth orbit, but that's another presentation. This says the same thing, but it has a cute little graphic. A friend of mine from the Czech Republic that I worked with put that together, and that just highlights where the red edge is. And it turns out that the red edge tells us a great deal about the amount of chlorophyll in leaves. And this graphic just points that out. Along the vertical axis is the amount of chlorophyll determined in a laboratory extract. And then along the horizontal axis would be the wavelength numbers of the red edge. And we call that the red edge inflection point. Inflection is where you look at let's think of a peak curve going up and then down. That point of maximum is the inflection point, and then you go from a rise to the inflection point to a decrease. And you can see that this R square that you see means that 87% of the spectral information is related directly to the amount of chlorophyll. And that's pretty good. Turns out that in the mesophyll, this is a diagrammatic representation of that, you've got the chloroplasts, but they're not absorbing at that wavelength, the near infrared. And so much of the light gets scattered, light coming from the sun gets scattered and then reflected back. And so this just says the patterns of reflectance in the near infrared, or the NIR, is influenced by the cellular arrangement of the mesophyll in the leaf. The state of health of the mesophyll cells will be reflected literally in that near-infrared region. And this is just a representation. You hopefully by now recognize the vegetation reflectance curves. Not much difference in the visible, but boy, look at the difference between healthy and damaged in the near-infrared. And again, we can see this from orbit. When chlorophyll is lost, it's called chlorosis. And this is an example of a leaf that has something called interveinal chlorosis, and that's a very good indication of stress. Where there is green, that's along the veins, and the area between the veins, the interveinal area, is yellow, no chlorophyll. You can see the same things. That leaf was an Angiosperm leaf, and this would be the needles of red spruce. This is actually from Camel's Hump in Vermont. Those of you that may be familiar with that mountain, I began my career studying vegetation health as an undergraduate at the University of Vermont, junior and senior year. And you can see yellow needles, and then you've got darker needles with chlorophyll. Okay, so leaf absorption, and this is interesting because it tells us how plants can tell time. Turns out that there can be flowers produced at a particular point in time, cones developed at a particular point in time, and then also leaf drop at a particular point in time. And this is just a schematic representation of the abscission layer, which is where the leaf, the base of the leaf, breaks off from the stem. This is an actual photograph showing a superized or a waterproof layer, and that's the separation layer below it. And that produces a superized leaf scar that we're seeing at this time of year on all of the woody plants. So here's how the plants tell time. They've got two different kinds of pigments. One that is very unstable in the light, and it becomes a more stable area in the dark. But when these chemicals see light, they actually convert back to the insensitive or sensitive layers. And it turns out that one decays to another or transfers to another over time, but that's in the dark. And so these pigments are called phytochromes, and they actually measure the length of night. Not the day, but the length of night, and that triggers the flowering process and the fall foliage display and leaf fall. Okay, let's talk about the wood wide web. When we walk in woods, we often see mushrooms. This happens to be a highly poisonous one, but an example of a mushroom. And they're in the woods because they're the mycorrhizal sexual reproductive structure. So this is just more information than we need at this point, but this is the life cycle of a mushroom. Here's a typical leaf. Looks like the diagram we showed before. And here would be the network of fungal cells. They're called hyphae. Those are the thin strands of a fungus, and they are throughout the soil. This is another example. The little fragments of things that are there are soil particles. The brown areas, those are the roots, and then that filamentous fungal hyphae connecting one root with another, and these could be different plants, a maple tree, an oak tree in combination. Turns out that a particular kind of a fungus that I've studied a lot is called an ectomycorrhizal fungus, and we'll abbreviate that EM. And it produces something called club roots. And this just shows the other kind of mycorrhizal fungus, an arbuscular mycorrhizae, that's abbreviated AM. The difference is the EMs don't enter the cortical cells of the root, whereas the AMs do. And you can see that structure called an arbuscle in the lower left-hand corner. This is a drawing I made way back in the early 1980s. Did a study in West Virginia looking at a particular kind of an oak called a chestnut oak. And the chestnut oak has the EM mycorrhizae, and you can see in the roots the club roots. Turns out this is an image of those club roots. This would be the arbuscular, the AM mycorrhizae, where the fungus actually enters the cells. But again, this is beneficial. This is not a disease. This is a beautiful example of an arbuscle inside the cell of the cortex of a root. This just tells us how many of the trees in the world are mycorrhizal, and you can see the large percentage, 72% are the AM mycorrhizae, and only about 2% are the ectomycorrhizae, but they all get along, and they all allow the roots of the trees to communicate with one another. So what are the benefits of having mycorrhizal fungi? I mentioned earlier the enhanced water and nutrient uptake, increased drought resistance, increased pathogen resistance, etc. And what does the fungus get? The fungus gets the chemical energy produced by photosynthesis in the roots of its host. I want to finish just by showing a particular study. It's a recent one. You noticed it was published in September of 2019. And the whole concept of a wood wide web is very recent. When I was in graduate school, we didn't know this at all. This is going to be a Gymnosperm that is studied in New Zealand. It's called a curry tree, and it can get to be 150 feet tall, can get to be huge, around 17 feet in diameter, just massive. And these trees live up to 1,500 years. Incredible. This just shows the leaves of the curry tree, and then another shot of the trunk. And I know this is a lot of data, but the important thing is off on the far left-hand side in either the day or the night is a stump. But that stump is living. The tree's been cut, but the stump is living. So it's got living cells in its roots, etc. And you can see that there are little blue arrows coming from the ground up into that stump, and then passing over to a living tree. And so that's a great example of water and nutrients being picked up by a stump that is no longer functioning photosynthetically, and it's sharing its water and nutrients with the host tree. And the graphic on the right-hand side during the daytime, that's the amount of water movement in the tree that's at the top, and you can see that dotted line is noontime. You can see in the afternoon the water movement is at its maximum, and look at the stump. It's exactly the opposite. So what's good for the tree is coming in large part from the stump. And the bottom graph just shows at night there's no photosynthesis, there's no movement of water, but again, that inverse relationship between the host tree and the living stump. Again, the idea of the connection between these, the living tree and the stump. If we had more time, I'd go into a description of what the graphics show. But the point is, the stump is sharing its water. It is taken up from the soil with another tree. And again, this just shows the stump and the tree, and they're in lockstep with one another. Tree is slightly higher because it's got leaves. The stump is lower. It doesn't have leaves, but they work hand in hand. You can actually measure that with something called a Scholander pressure bomb, and you put a leaf with its petiole sticking up out of the top. You put in some compressed air, often it's a nitrogen gas, and you can actually force the water in the leaf out through the end of the petiole, and that tells you how much water was in the leaf and how much tension the water was under. And this is the last slide. It's my own Scholander pressure bomb in my lab. And here I'm using a little magnifier to look at the cut end of the petiole to see the water coming out. So what have we learned? Well, we learned that trees' growth rates are affected by their environments, both physical and chemical. Plants can tell time. They know when to flower, when to drop their leaves. Trees can help one another by sharing water, nutrients, and chemical energy. And then trees can communicate with us through their leaves by using spectral reflectance properties. So I'd like to think of myself as the Lorax. If any of you read to your kids or your grandkids about the Lorax, you know that the Lorax says, "I speak for the trees, for the trees have no tongues." Well, the first half is important, I speak for the trees, but trees do have tongues, we call them leaves, and they tell us about the health of the trees. So I want to thank you very much. We're going to be looking at the Earth from space in the next presentation, and I hope you have learned at least one new thing about trees. So thank you.
And we're back live. So I hope you enjoyed that Secret Life of Trees presentation that Professor Barry Rock gave. I don't see any questions yet on our screen, but our Forest and Climate Change Subcommittee of the Social Justice Committee has created a few, and I'd like to read a couple to you, Barry, if I may.
Great.
The first one is, would warming of the climate increase the rate of photosynthesis? And if so, would that offset the loss of the number of trees?
That's a great question. It turns out that as the climate warms, increasing temperature doesn't affect the rate of photosynthesis. That is all determined by enzymes within the chloroplast, within the thylakoids, the light reaction, the dark reaction, the coupling of carbon dioxide into making sugars, which then end up making cellulose, cell wall material, and stored. But it's enzymatically controlled, not temperature controlled.
The second question, what species of trees at our latitude are most likely to survive climate warming?
That's a good one. I think our oaks will survive, at least for a time. Back during the last period of glaciation, areas here in New Hampshire, in New England, all the way across to Kansas and that sort of thing, they were covered with 2 miles of ice. And so we didn't have any trees. And this would have been 12,000 years ago. Trees to the south of the furthest extent of that continental glacier got down into Kansas, but it turns out the southern states, what would become the southern states, had a lot of oaks. And then as the ice melted, and that happened very quickly, within a matter of maybe 100 years or 200 years, trees began to migrate north. Our sugar maples, some of them survived in little enclaves when the ice was here. But the oaks we have today were actually from the south, and they migrated north. Since they are southern species, the oaks will likely tolerate warming temperatures. Our sugar maples, unfortunately, they'll move north. They'll end up in Canada. And some of my research shows that sugar maple is currently under stress from the warming climate that we've had, and it could get ever so much worse. And so those sugar maples will try to adapt, but most likely they will, over time, just migrate to the north. Our birch are very hardy, so they'll likely survive. So I think the oaks and the birch are likely to survive. Maples, probably not. And that's too bad, because I think when we think of New England, especially Vermont, New Hampshire, Maine, we think of maple syrup.
Right.
And it's sobering to think that our maples, are iconic New England sugar maples, are under stress right now, and they're not likely to survive here.
What's the time scale you're referring to in terms of surviving? Are we talking 50 years, 200 years, 1,000 years?
That's a very interesting question, because just yesterday, March 1st, the Intergovernmental Panel on Climate Change released their most recent findings. And probably the finding that is of most concern to me, and should be to everyone, is how quickly things are happening. And in your introduction, you mentioned the New England Regional Assessment. That was published in 2001. And in that, we had models projecting what's going to happen over the next 100 years. And we thought the sugar maples would be stressed over the next 100 years. But 2001, that was 20 years ago, and already the maples are under stress. So things are happening much more rapidly. So it's really difficult to put a number of years. On the other hand, time is growing short for us to do anything. Yeah. And it's really important that we address the question of climate change. Trees are there doing that for us for free. Carbon dioxide being absorbed, converted into cellulose, stored as cellulose. I mean, it's really amazing what trees are doing. And if the maples die out, as they will if it gets much warmer, then they'll still be surviving in Canada, and the oaks will be able to tolerate the warming. So we're not going to lose all of our trees. We just will lose certain trees. And I think sugar maple is one of those types of trees that is likely to be impacted.
One other question that follows on with that is this person is asking, what species of trees are most efficient in addressing climate change?
Well, you need a combination. You need fast-growing trees. Young trees growing rapidly are absorbing a lot of carbon. Older trees, often what we would call old-growth forests, are absorbing carbon, certainly, but not at the rate of an actively growing young tree. And so the kinds of trees that we should think of planting would be fast-growing trees. And what comes to mind, interestingly enough, is a tree called the tree of heaven. And we don't have it here in New England yet. But I would guess within the next 20 years it will be here. It's called the tree of heaven because how fast it grows. It's like it's growing to heaven. And this is Alanthus altissima, and altissima means high. So my guess is that could be a super tree because it grows very fast. In a year's time, a sapling can grow 3 feet.
Wow.
So, I mean, it's growing very, very fast, getting very, very tall, getting very, very thick. So it's storing a lot of carbon. And so that may be something we want to think of. We also want to think of trees that have very dense wood, because that's got a lot of carbon stored, a lot of cellulose, very thick cell walls, vessel elements, fibers, tracheids, that sort of thing that we talked about in the presentation. And black locust is a very good tree. It has very dense wood. It's good for making fence posts.
I recall from my dendrology class a type of tree called ironwood.
Oh yes, yeah, Carpinus.
My God, that was just you could hardly move the thing in a 5-inch diameter stem.
Oh yeah, no, and that would be a good tree then. So thanks for bringing that up.
Yeah.
The oaks, they have very dense wood, and they come from the south anyway. So as it gets warmer, they're going to say, this is more like it. So we've got some trees that are here now, and if we planted more of them, that would be a good thing. But keep in mind that we want fast growing, so maybe the tree of heaven would be one. Dense wood, Carpinus, the ironwood, keep the oaks that we have, black locust.
Yeah.
So we've got to get clever about how we address this issue, especially using forests as an ally in the fight against climate change.
Looking at the animal kingdom for just a moment, 30 years ago, maybe 40 years ago, I never, ever remember seeing a possum in New England. And they're all over the place now.
And they're a southern species.
They're a southern species.
And they've been working their way north as the climate warms and that sort of thing. Yeah. And growing up in Vermont, we would be out in the morning at 8:30 as soon as we were done breakfast, and then would come in at probably 4 o'clock, 5 o'clock in the afternoon. And this was during the summer. We were playing every day. We never saw a tick.
Interesting.
They just were not in Vermont.
Yeah.
And that would have been back in the 50s and 60s.
Yeah.
And now you can't walk your dog without doing a tick check. So, things are happening very quickly, just as the IPCC report indicates.
Yeah. Well, I want to thank you very much for helping inform us all about trees and their secret lives. And I remind people that the third and final presentation next Wednesday evening, March 9th, will be on remote sensing of forest health from space, from orbit. And we hope you can join us then. And in any case, this session, as well as the previous one last week and the one next week, eventually will be shown on cable channel 98, seen by Exeter residents, and some of the programs can also be seen by channel 13 residents in the surrounding 6 towns of SAU 16. In addition to that, the videos and the whole presentations will be uploaded to the New Hampshire Coalition for Community Media TelView server so that other communities can download that and then broadcast it to their citizens over their local cable system. And they'll also be shown on Exeter's YouTube channel. So there'll be many opportunities to see these if one missed the live presentations on the dates of February 23rd, March 2nd, and March 9th. Thank you again. Maybe we'll see you next week. Thank you.